Okay, hello everyone. It's 11 o'clock Central Time, and I'm glad to see that our participants are chatting in saying they can hear fine. That's excellent news. We'd like to thank you for joining us for today's webinar entitled Whole Genome Amplification, or WGA, What to Do When You Don't Have Enough Genomic DNA. My name is Beth Fry. I'm a product manager here at Lucigen, and we just wanted to welcome you all. And before we introduce today's speaker, I have a few housekeeping notes for you. First, please note that all the participants are on mute, and you'll remain on mute for the remainder of the webinar. If you are on a mobile device like a cell phone, we ask that you mute that device to make sure there's no background noise. We want to make sure that we answer all of your questions on WGA today. We're asking you to hold your questions until the end of the webinar. We should have time for questions. You'll notice on the left-hand side of your screen there's a chat window, and we ask you use this chat function to ask questions at the end of the webinar. And also, please make sure that the little button at the bottom of the chat window is set to say all, so this will ensure that we can see your question. Also, during the webinar, we'll be asking some interactive poll questions, which is an opportunity for you to tell us about your experiences with whole genome amplification. And I'll be taking over from our speaker in order to ask the poll questions. And you'll have an opportunity to vote live using your computer mouse, and we'll all see the results. So we hope you enjoy the polls. We think they're a fun way to make the webinar a little more interactive for everyone. And please note that if you're watching the webinar in full screen mode, you'll have to exit full screen mode in order to take part in the polls or to ask questions on the chat window. So feel free to watch the content in full screen, but you'll have to hit the escape button to exit full screen in order to interact with us. So okay, that is all for housekeeping notes. And now we are very pleased to introduce today's speaker, Rob Brazos. Rob is a senior product manager here at Lucigen. He has a wealth of technical bench experience in genomics, molecular biology, and protein biochemistry, along with array analysis and next-gen sequencing. And we are excited to have him presenting today's topic, Whole Genome Amplification, or WGA, What to Do When You Don't Have Enough Genomic DNA. Take it away, Rob.
All right. Thank you, Beth. The senior just means that I'm old. We'll start off with a joke. All right. So agenda today, pretty straightforward. What I'd like to do is first talk about what whole genome amplification is. It's pretty straightforward, but we'll talk a little bit about the nitty-gritty of what it is and what it means. And then we'll get into some of the challenges of poor whole genome amplification. What does that mean for you in terms of the experiments that you want to do? Then we'll delve into the different methods of WGA. So we'll review PCR methods and MDA, or multiple displacement amplification methods. And we'll talk a little bit about the strengths and weaknesses of each of the two groups. And then we're going to focus in on MDA. And there's quite a few reasons for that we'll show you and show you data to support those reasons as well. But we're going to talk about a variation on MDA called the Sygnis TruePrime WGA kit technology. This is pretty cool. It's pretty straightforward. And I'll show you what the differences are between standard MDA and the TruePrime kits. Then I'll give you some overview of what you can expect with those kits, a little bit of workflow, a little bit of yield data, contaminant data, things like that. And then we're going to delve really deep into some sequencing results that were recently published in a Nature Communication paper from our friends at Sygnis and colleagues. And they've given me the go-ahead to talk about that data today. So that's going to be a lot of the real data that we're going to look at the end. Then, of course, we'll close with a summary and some final thoughts. Alrighty, so on to WGA. So this slide just gives you an idea of what whole genome amp is. It's not very complicated. The idea is here you've got a very simple situation where you've got two homologous chromosomes. They might have different alleles at particular genes. So here's a heterozygous set of alleles, another heterozygous set, and here's a homozygous allele at a different gene as an example. Now, if whole genome amplification went perfectly, you would copy both of those homologous chromosomes equally, and you'd copy them the same number of times. So you'd have, in this case, 10 copies of homologous chromosome number 1 and 10 copies of homologous chromosome number 2. Now, why would you want to do that? There's many reasons. So first, you might not have enough material to do your experiment. So if you're starting with a single cell, you might not have enough DNA there to do the type of experiments that you want. You just don't have the sensitivity. So you can amplify that material. The other thing is that you might have a technology, there might be a single cell sequencing kit out there that you could use. But once you use it, you've used up that single cell and you have nothing to go back to. Okay? So with WGA, you can amplify that material, then do your sequencing, and still have enough material to archive and go back to later for additional experiments if you need to do that. So you have a repository, which is really beneficial. And as I kind of mentioned, there's lots of different sample types that you might be amplifying from. So it could be single cells. Let's say you're looking at a tumor sample and you want to look at the different cells in the population. You want to see what kind of genetic variation they have within the cells. You could do that for cancer, or cells that you've sorted, whatever the case might be. Or you might have limiting amounts of purified genomic DNA. So maybe you had a biopsy sample, research sample, and you extracted DNA from that, but you have very little. So you might want to amplify that as well. Or let's say a metagenomic sample, like a bacterial genomic DNA from dirt or fungi from dirt or something like that. There's very little DNA usually in those cases, and you want to probably amplify it before you analyze it. So that's what we're going to talk about today. So the first poll question, here it is. Beth's going to take it over, but what type of samples are you or will you be interested in amplifying with WGA?
All right, let's bring the poll question window over here. We're going to open the poll, and we would like you to know what kind of samples you're using. So you can go ahead and vote using your computer mouse. Are you using fresh or frozen tissue from biopsy, so purified genomic DNA, FFPE samples, metagenomic samples, single cells? Look like there are some people using single cell samples, microbiome samples. If you do have a sample type that is not listed here, please click Other, and then let us know in the chat window what your sample type is. And then we have a couple people that are no vote. They may not be doing WGA currently, but maybe thinking about it, just learning about it. So it looks like the winners so far are single cells and fresh or frozen biopsy tissue. All right. I think if everybody's had a chance to vote, we'll go ahead and finish this poll and hand it back to Rob. Thank you, everyone.
Interesting. Yeah, we did an AIM webinar this morning too, and that's pretty similar results. It looks like more folks are down there, not quite sure what they're going to do yet, but the metagenomic and microbiome samples were more popular this time. So it's kind of all over the board. It's basically whenever you have limiting DNA, you need to amplify it. So, all right, that first slide I showed you about whole genome amplification was basically a perfect situation, right? If you do your whole genome amp and you get 10 perfect copies of each homologous chromosome in this example, that's awesome. But we all know that nothing works that way in research. And so when you do real life whole genome amplification, certain things can happen. So I'll just touch on it briefly here. We'll do a little bit more in the next slide. But you can have areas that don't get amplified. You can have areas that amplify a little bit but not all the way. You can get alleles that are converted from heterozygous to homozygous. All of those things are bad. And so we're going to talk about ways to improve that today. So hopefully in the end of this talk, you'll have some choices as to what kits to use to improve your whole genome amplification. So what are those differences in a little bit more detail? So in a perfect world, you'd really want uniform amplification of the entire chromosomes. Okay, that's both in their representation within the sample and amplification of the whole thing. If something is missing, you won't get any data from that area, right? No matter what your assay is. So you need to have uniform amplification of the entire chromosomes. You also want accurate representation of each set of alleles. So if you've got heterozygous alleles, you want them to stay that way. You want them to continue to be represented in the same frequency in the amplified material. If you've got homozygous, same thing. You want them to remain homozygous. That's a little bit easier. That's not as much of a hard. The bigger problem is converting heterozygous alleles to homozygous alleles. You also want high fidelity amplification. So a lot of times people want to look at single nucleotide variants or insertions and deletions. You don't want your amplification method to introduce any of those types of changes. So you don't want any SNVs being created by, like, let's say, error-prone PCR or something like that. And so that's all, again, in a perfect world. The bottom line is the amplified genomic DNA equals the starting genomic DNA, except there's more of it. Okay? Now, in real life, as we said, things go wrong. Right? First of all, there's uneven amplification across chromosomes. So there's missing areas, as illustrated here, just didn't amplify. There's areas that are unequally represented. So here there's some representations of area that didn't get replicated a little bit, or amplified, and here it did get amplified. There's examples of loss of heterozygosity also. So heterozygous alleles changing to AA. So here you had a light green and a dark green allele, and now in this sample, everything's dark green. Okay, so you've lost that heterozygosity, and that's obviously not something you want to do if you're looking at cancer genes or something like that, and you have different alleles. You want to maintain the frequency of those alleles in your sample. Let's see what else. Introduction of contaminants. We'll talk about this in some detail, but these different methods, they're very sensitive. And you can either amplify environmental DNA that you don't want in there that gets contaminating into your sample, or you might actually get artifacts introduced by the amplification method that you're using. And we'll show information on both of those today. You might also create chimeras where you get jumping or fusions that aren't natural. So here there's an example. Those might get introduced. And the bottom line is the amplified genomic DNA does not equal the starting genomic DNA perfectly. It's not a perfect representation. And that's just biology, and that's the way molecular biology works. But what we're going to talk about today is a way to help you improve that, to make your amplified material more closely resemble the unamplified material. So when you do have those issues, what are some of the problems that might create for you? It's pretty straightforward, right? So if you have incomplete or poor amplification, you're going to get, if you're trying to do whole genome sequencing, you're going to have incomplete whole genome sequencing results, right? You won't have the whole genome represented, so you can't get the sequencing information there. That might lead to the inability to assemble whole genomes. And also, if you have contaminating sequences, it might make it more difficult to assemble whole genomes. If you're looking at maybe, let's say, a metagenomic sample, you have different species in there, and they have different abundance, and you want to maintain that abundance or representation. If you have bias in your amplification methods, you might skew the results of that metagenomic sample. And then finally, you may make inaccurate calls or difficulty identifying single nucleotide variants, copy number variants, and or structural variants. So let's say a BCR-able fusion or something like that. If the amplification doesn't work well or high fidelity, you might have difficulty doing any of those different applications with the whole genome amplified materials. So that brings us just to poll nut question number 2. Just a little break here. Beth, take it away.
All right, so our question is, what do you plan to use amplified samples for? So what do you want to use whole genome amplification for? Do you want to use it to do de novo whole genome sequencing, resequencing for copy number variant analysis or SNV analysis, microarrays? Maybe you want to identify some organisms in metagenomic samples, archiving precious samples if you have samples that are very precious to begin with, very low amounts of DNA, PCR analysis, or other. If you do have another application that's not listed here, please let us know via chat. It looks like a majority of people are doing whole genome sequencing and PCR analysis for specific genes or specific loci. All right. All right, well that's wonderful. That's great information for us to know what our audience is doing, and thank you. I hope you enjoyed that one. We'll finish it here and then hand it back to Rob.
All right, de novo whole genome sequencing. So can anybody chat quickly in the chat window what type of samples you're doing the whole genome sequencing on? That one surprises me a little bit. I'd just be curious to capture that information in the chat window. But in the meantime, I'm going to keep going. Okay, all of the above, okay. Or cell-line authentication, oh, cool. All right, so now let's switch over and talk about the different methods that are available. I'm going to start with PCR-based methods. These are basically obviously PCR-based, but they use different primer designs to try to do whole genome amplification. So the first one's called linker adapter or linker anchored PCR. This is really similar to making a genome sequencing library these days and then doing PCR amplification of it at the end. So you start with genomic DNA, you'll shear it into correct size fragments, and then you'll ligate on adapters to each end. And in this case, the adapters are embedded. They have a sequence within them that will be a landing site for PCR primers, specific PCR primers. And so then you will add those primers and you'll amplify by PCR. So again, very similar to a lot of NGS kits right now. IRS PCR, it is not the Internal Revenue Service in the US. It is interspersed repetitive sequence PCR. So these are, it's similar to the one just talked about, except there's no ligation. what you do is you design the primers to land in known repetitive elements and then amplify by PCR. So this one, obviously, you can see right away. If repetitive elements are too far away, you're not going to amplify the region in between because PCR has limitations on the length that it can amplify, and that'll quickly introduce some bias there. Finally, PEP PCR, primer extension PCR pre-amplification PCR, excuse me. This uses random 15-mer PCR primers. They're kind of degenerate. And they're used to amplify the genomic DNA under permissive conditions. So the annealing temperatures and extensions are lower so that you get sort of random PCR across the genome. I should also mention that these figures and stuff were pulled from Blaney in paper in FEM's microbiological reviews. So there's a lot more information on single cell work and different amplification methods. So if you're interested, I'd highly recommend you check out that review where I pulled this information from. A couple more PCR methods. There's DOP PCR, or degenerate oligonucleotide primed PCR. So in this case, the primers have degenerate 3' ends. In fact, actually, they have about 6 bases that are constant. And then there's a section that is degenerate. And then at the 5' end, you have a constant region. Okay, so it's kind of a mixed bag. You need a little bit of specificity at the 3' end to help with priming, but then you have a degenerate sequence to force things to land all over the genome, okay, or promote things landing all over the genome. So then you do primer extension at random sites across the genome at low temp annealing, and then you raise it to higher temp. So things that amplified will have the primers in them, and you can use those same primers for PCR amplification at higher, more stringent conditions. A more recent development on DOP PCR is DDOP PCR. These are just fun to say. This is displacement degenerate oligonucleotide primed PCR. Okay. This one's a little bit different. It's closer to MDA that we're going to talk about in a second. But again, the primers have degenerate 3' ends and constant 5' ends. Okay. And you do an extension reaction, primer extension reaction at random sites at low temp annealing, promoting sort of uniform annealing and amplification. But you use a different enzyme, one that's got strong strand displacement capability. So you will amplify, you will displace strands, you can displace original strands, you can displace newly synthesized strands, the primers can land on newly synthesized material, and you'll end up getting fragments that have the primer binding sites on both ends. So then you up it to PCR amplification conditions at higher temps, and you spike in new PCR primers that will recognize those constant ends, and again, amplify the material here. So that's all the PCR methods. Now we're going to talk about multiple displacement amplification, or MDA. And this is really, as I just said for DDOP, this uses an enzyme or DNA polymerases that have strong strand displacement activity. That's one of the key hallmarks of MDA, and you'll see why in the next couple of slides. So standard MDA is illustrated here. You start with genomic DNA, you denature it in the single strands, you add random hexamer primers that can bind anywhere, and then you will extend those with DNA polymerase with that strong strand displacement. So again, you'll start synthesizing, you'll displace old strands, you'll land on new strands, start synthesizing, displace new strands, old strands, whole 9 yards. You'll get these big masses of amplification going on. And the key point here is this is a little easier than PCR because it's isothermal. So these will run at standard reaction temps, like single reaction temps, like TruePrime will run at 30 degrees, okay, for instance. There's also another variant of this available. I won't talk about it, but it's based on the T7 phage replication system. You can get more info here if you're interested. And then second to last one is SPIA, or single primer isothermal amplification. This one's a little bit trickier. Basically, you use primers. They have a specific RNA sequence fused to a partially degenerate DNA primer sequence. So the 5' ends are RNA, the minimal 3' ends are DNA. And what you'll do is you'll add those to samples, you'll get primer extension at a set temperature, and then you add in RNase A. So any RNA-DNA hybrids, the RNA portion will be degraded, and that allows other primers to infiltrate and to do reinitiate extension. So you have another strand displacing enzyme again, and you can start synthesizing more and more strands off of here. Okay, and then finally, there's a new one. It's called Malbec. If I say Malbec during the presentation, I apologize. It's one of my favorite wines, but It is called Malbec, which is multiple annealing and looping-based amplification cycles. So here you go. I'll try to explain this one. You start with genomic DNA. You'll add degenerate 3' ends primers again, constant 5' ends. They'll anneal. You'll do primer extension, sort of quasi-linear amp at random sites with thermal cycling. So you can go up and down with the temperature, and you prime and amplify a lot of different regions from single primers. And these primers are designed so that when you end up with primers at both ends of a fragment, they'll loop on each other. And there's actually some benefits to that. I won't go into that in great detail, but there are supposedly benefits to that in preventing weird stuff from happening, chimeras and things like that. But again, I suggest checking out the original paper and getting more information. Then what you'll do is you will then shift up to with high temp. Those things will unfold, and the primers will now PCR amplify the final material. Okay, so we're going to switch and really talk about MDA today. But before we do that, I just want to tell you why. So these are the perceived strengths and weaknesses of the two groups, both perceived and real. Some of it's been proven, some of it's general perception of PCR versus MDA whole genome amplification. So amplified links, you tend to get big fragments with MDA-based amplification. PCR is small. We all know that PCR favors smaller fragments. It's just the bottom line. Nucleotide error rate, MDA usually uses Phi 29 DNA polymerase, which is a very high fidelity enzyme, 10 to the -7th, 10 to the -8th error rates. Very good enzyme. So you get fewer errors. PCR-based, depending on the polymerase you use, 10 to the -4, 10 to the -5. You can improve that with higher fidelity, but that's generally what occurs. Chimera formation, we talked about this earlier, believed to be higher with MDA and lower with PCR-based methods. Completeness of genome coverage. This is one of those keys, right? If you're amplifying a genome, you want to amplify it all. If you're doing a WGA type method, you don't want to amplify some regions like you might with PCR. You want to amplify the whole thing. So it's believed, and I'll show you data, that MDA does a very good job of amplifying the whole genome. And it's a little bit, doesn't do quite as well with PCR-based methods. Variability. It's believed to be high with MDA and low with PCR. I'm going to show you data with some of the kits that we're talking about where it's actually quite good and very reproducible. CNV detection, or copy number variation detection, generally believed to be poor with MDA and good with PCR. And this has to do with the uniformity of coverage, or uniformity of amplification and then sequencing coverage. And you'll see that, in fact, we've got great data with some of the MDA kits today. Duplicate formation, lower or higher, not going to talk about that too much. Allelic dropout, we talked about that. It's believed to be about 5 to 50% for MDA, and it's kind of unknown for PCR-based methods. SNV detection, single nucleotide variants, good with MDA, plus minus with PCR, so it's okay. And the protocols, since the MDA-based methods are generally isothermal, fewer steps, they're pretty easy. PCR-based methods can often be multi-step, as you could assume from the methods that I just showed you, the flows in general. All right, so that brings us to our last question before we get into the meat of the data. So Beth, take it away.
All right, let's open this poll for you all. And this poll is what type of data is valuable to you when interpreting performance of WGA kits. So here, we'll quick show results to everybody so you can all see the results. Do you care about coverage breadth or depth or uniformity? Do you care about a comparison to unamplified controlled genomic DNA, chimera rates perhaps, zero coverage reasons? Do you like to see comparisons to other methods or other kits available to you? Or application data, is that important to you? Or maybe you're not sure what sort of data is important to you at this point in time. That's okay too. If you have something else that we haven't listed, again, please type it into the chat window. That would be great. Looks like most people in the audience today are worried about uniformity and the breadth of coverage, making sure they get all of the genome and the uniform coverage. All right. I think we'll close it out there. Thank you, everyone, for your participation in our polls. That's our last one today, so we'll have Rob continue on with the slides.
Yeah, that's perfect. I think that's, I think everybody hit it right on the nose. like I said, if you don't cover the regions, you can't get data, right? No matter what assay you're doing, if you don't cover them uniformly, more difficult to interpret the data. So you're going to see a lot of coverage data today in the rest of the talk, so that's perfect. And then we'll get into some application data showing what the benefits are of uniform coverage and breadth of coverage. So we're going to focus on MDA, and that's exactly, and the reason is because of the completeness of that genome coverage that I just alluded to in the previous slide. It does a really good job of amplifying genomes and amplifying significant portions of those genomes. Okay, we're going to talk about different kits and methods today. We're going to talk about Repli-G. This is a commercially available MDA kit from QIAGEN. It's very popular. It's been used a lot. This one uses random hexamers as illustrated in the method figure I showed you a few slides ago. Okay, then we're going to introduce TruePrime. These are WGA kits that are also MDA kits. They're made by Sygnis. We sell them, and they have a single cell and a general purified genomic DNA version as well. But these are different MDA kits in the sense that they don't use random hexamers, or random primers. What they use instead is what I'm going to simply call a primase enzyme to synthesize primers on the denatured genomic DNA instead of having random primers floating around. And you'll see there are significant benefits to that. Then we'll talk about generic MDA experiments. When you see this, this is the TruePrime kit, but we've substituted in, or Sygnis has substituted in, random primers in place of the primase. So that's a direct comparison to the effects of random primers versus using primase. And finally, we'll include Malbec experiments here. These are is a single-cell Malbec kit available from Yicon Genomics, which is again a commercially available kit. The majority of the data that I'm going to show today is published in a recent paper by Angel Peter. He's at Sygnis and a lot of other authors on this paper, but it's in Nature Communications. If you have any questions about the data that I can't answer, I highly recommend you dig in there and dig into the supplemental data. There's a lot of good supplemental data in here, and I'm going to show quite a bit of that today. Some of the data was also just provided to me by Delucigen by Sygnis, and we'll present that as well because I think it's really informative as to what's happening with random primed MDA. All right, so I told you that TruePrime was different. Here's how it's different, okay? Basically, they've identified this enzyme. It's called TTH-PrimPol. It's a primase and a DNA polymerase. We're using it for the primase function. Basically, it binds to the denatured DNA in your sample when you're trying to do your WGA. It synthesizes short primers. These are then, the enzyme is then displaced by Phi-29 DNA polymerase that's in this mix, and it begins synthesizing long strands of copies of the genomic DNA. Okay, and it has strand displacement activities, as we said. So as it encounters another double strand, it can displace that and keep going. So that's a benefit to getting uniform and significant coverage of these genomes. Then, of course, primase can land on anything that's single-stranded. So it'll land on these newly synthesized strands. It'll make a short primer again. Phi-29 DNA polymerase will displace and then amplify those primers as well. So you get this big mass, this big clover leaf or flower, whatever you want to call it, of amplification happening in the tube. Okay? So again, the key difference is using primase instead of random hexamers, random DNA hexamers. And here's just the protocol. There's two kits, as I said. Let's look on the right one first. This is the TruePrime WGA kit. So this is when you have purified genomic DNA. You basically add it to a tube, add some buffer D, which denatures the DNA. It's an alkaline denaturation. Let it go for about 3 minutes at room temp. Then you add buffer N to neutralize, bring it back down, neutral pH. Then you add your amplification mix, incubate at 30 degrees for 3 hours. You can go up to 6 hours. You can go lower than 3 hours if you want. But we really recommend 3 hours appears to be the sweet spot for most amplification reactions. Then you've got all your amplified material. You just have to heat and activate at 65 for 10 minutes to kill the primase and the Phi-29 polymerase. And you've got your amplified genomic DNA ready to go. The single cell kit, very similar except for the first 2 steps. So in this step, you take your single cell in PBS. You add buffer L2 to it. L2 is a lysis buffer to lyse and pop the cells, and in general mammalian cells here. And then also to denature. So it's got sodium hydroxide in there as well. Incubate at room temp for 5 minutes and then 1 minute at 95 degrees. And then you add buffer N again to bring the pH back down. and then you follow the same protocol with the amplification bits, and then heat kill. Okay, so very straightforward protocol, single tube, very easy to follow. Here's some of the data you might expect. So, we talked about size a little bit in terms of products. Here's an example of the difference between some of the different methods. So Sygnis tested Malbac, TruePrime, and the Repli-G. And what you can see is Malbac, as expected, produced smaller fragments because it's kind of a PCR-based MDA method, right? It's kind of a hybrid. So here that they said the fragments were about half a kb to 1.5 kb in size. TruePrime, significantly bigger, about 1.5 to 12 kb. And then Repli-G looks quite a bit bigger. Now what's interesting is you see this kind of discrete band right here with the Repli-G. I actually expect that might be some junk. And I'll show you why I'm expecting that. I don't know for sure. We haven't tested it, but I suspect that that's some garbage that got amplified in these samples. And I'll show you why that in a couple slides. So the yield. This is important, right? You're starting with limiting amounts of genomic DNA or single cells. This is an example of using the TruePrime WGA kit versus the generic one, which is TruePrime with the random primers inserted instead of Primase. And so what you can see, the no template control is critical. This stuff will amplify anything. Remember, it's not specific because there's no specific primer design. So you want to make sure you do a no template control. And then you can see in blue that the TruePrime WGA kit was able to amplify material from as little as 100 nanograms. Pretty small error bars, and it increased with the increasing amount up to a nanogram. Okay, this is again using human genomic DNA from Primase. And it's important to point out here that the quantitation was done with Pico Green from Thermo Fisher and Vitrogen LifeTech, whatever their name is these days. And what's important here also is that this is basically a double-strand specific DNA fluorescent dye. So this is only amplifying material that's double-stranded, and that should sequence well. Okay. You can see the TruePrime with the random primers instead of Primase actually didn't amplify as well, much lower sensitivity. You really didn't start to get any material until 100 femtograms. So this kit with the Primase is about 100 times more sensitive than the same kit with random primers. So that's going to be beneficial depending on how much starting material you have. If you have very limiting amounts, this is going to really help you out. All right, now let's look at some of the artifacts. So this is really one of the big differences between these two types of kits. Random MDA with random primers versus MDA with Primase. So on the left is the TruePrime WGA kit. Again, 1 picogram of input genomic DNA from Primase. And then on the right is the generic results from random primers. So in this experiment, 1 picogram of input, as I said, TruePrime WGA kit and protocol, or on the right, substituting Primase with random primers. So it's again a direct comparison of the two kits, two technologies. And then the amplified DNA was made into sequencing libraries, NGS libraries, sequenced, and then mapped back to known genomes. Excuse me. You can see on the left that over 99% of the sequencing reads mapped back to human. So the target genome, what you expected to amplify, was what you got in the sequencing results. On the right-hand side, you can see that only about 75, 76% of the DNA mapped back to human. And the other stuff is a whole bunch of unknown DNA, invertebrate DNA, and then some synthetic DNA. I suspect that this unknown DNA is concatenamers of these random primers. They just sort of land on each other. They start to get extended, and you can end up with this big mass or amplified material that really doesn't correspond to anything. Okay, and I think that's what you might be seeing in those large Repli-G band that was very high and very kind of bunched up there at the top of the gel. Not proven, but obviously the more information you have from your target genome, the better off you are, right? You're not wasting valuable, if you're doing sequencing, you're not wasting valuable sequencing space. All right, and then just to show, that one of the key take-homes with MDA-based applications or amplifications is that as you decrease the amount of input genomic DNA, you're going to start to see a little bit more of the contaminants. It's higher likelihood that they'll start to pop up in the analysis and be amplified because they're not swamped out by significant numbers of copies of the target genome. So here's just an experiment where they used a TruePrime WGA kit with 1 nanogram input human, 1 picogram input human, and 1 femtogram input human. They did the same kind of experiment. They amplified for a long time, 6 hours, and they subject it to next-gen sequencing and mapping again. And you can see that everything's great here, kind of like what I just showed you. Even down at 1 femtogram, over 95% of the sequence reads map back to the human genome again. So it's still, you're not producing this garbage at a very high frequency, even with as little as 1 femtogram, which of course doesn't even represent a whole genome from a human cell. So pretty cool. Nice technology. Again, very little garbage introduced in the amplified material. Now here's the final one, and this is kind of a unique attribute of primase-based MDA. So in this experiment, we started with a picogram of denatured human genomic DNA from Promega in 2 WGA reactions. Okay, one is the TruePrime, and one is TruePrime with random primers instead. And after neutralization, 1 nanogram of non-denatured yeast genomic DNA was added. So this should be double-stranded, should not be a template for MDA because it hasn't been denatured. It was added in after the denaturation and neutralization steps. Okay, so the idea here is to see, well, how much of that yeast genomic DNA is actually amplified. So then they were run through the rest of the protocols, again, sequenced and mapped back to the human genomes. Okay, and what you can see is that on the left with the TruePrime kit, only about 7.5% of the sequencing reads were yeast genomic DNA. A little bit of unknown stuff too. On the right, in contrast, when you used random primers, it went up to about 54% of the reads corresponded to yeast. Okay, and I've been told, Angel told me that basically they believe that Phi-29, if a random primer can get into the genomic DNA somewhere, it will amplify. And that's why, even though this was denatured, there's probably breaks and overhangs and things like that. The random primers can get in there well enough to amplify that material extremely well. Okay, and that's why it overtook the number of human sequencing reads in this target sample. Excuse me. So again, Primase doesn't have that ability. It needs a little bit of a bigger landing pad to go do its thing in terms of making primers for Phi-29 to extend. So again, another benefit of using a primase-based MDA method. Okay, so we're going to switch over to, that's kind of the general stuff about the MDA kits, a little bit about the specifics of the TruePrime MDA kit. I want to now dig in deep into this paper that Sygnis published in Nature Communications. And I want to look really hard at sequencing results from WGA material produced from single 293 cells, okay, using various kits and methods. So it's the same kits I mentioned earlier. We're going to do TruePrime single cell kit, the generic MDA, which is the TruePrime kit with random primers. We're going to do Repli-G because it's super popular and it's from QIAGEN. And then we're going to do the Malback kit, single cell kit, which is also commercially available. We're going to make amplified material according to everybody's protocols, of course. We're going to shear it using a Cavaris focused ultrasonicator, or they did shear it. And then they constructed libraries using the NAB Next DNA library kit. And I just want to include here this little red call out here that this kit includes a PCR step at the end. So you make the library and then you PCR amplify as well. And of course, you have potential for bias with WGA and you have potential for bias with PCR. And we'll bring that up right at the end as a closing thought for these types of experiments. So then the libraries were sequenced deeply on a HiSeq 2500 using 2x125 base pair paired end chemistry, version 4 chemistry. And then the sequencing results were sampled. So they took samples of random reads, 12 million and 5 million in the experiments I'm going to show you. And they analyzed coverage, breadth of coverage, depth of coverage, all the data that I'm going to show you was analyzed and then done all, of course, based on the experimental goals of each of the figures that I'm going to show you. And just a reminder, most of this data was pulled from this Nature Communications paper. So please go back if you want more detail and check it out there. Oh, I should also point out that you're also going to see NA or non-amplified as the control. In that case, that's taking 293 cells, multiple cells, making genomic DNA from them, and then constructing libraries from that non-WGA material. So this is going to be the non-amplified control sequencing results. And that's sort of the gold standard in this case. All right, that's the control, that's the gold standard. So here's the first slide. You can understand this fully. These figures are hard to look at. So we're gonna dig in, we're gonna zoom into some of these chromosomes in a bit more detail. But in this experiment, basically they did 5 different types of samples for sequencing: the non-amplified control, TruePrime, Repli-G amplified material, Malbac, and the generic MDA kit. And the colors match up to the colors in the circles. Okay? They analyzed 12 million read pairs and then found the average coverage in 50 kb bins. So they moved along the chromosomes in 50 kb jumps, averaged the coverage in each, and the height of the bar, height of the lines indicates the coverage, and the scale is 0 to 2500. Okay? So outside ring, non-amplified, blue is TruePrime, orange is Repli-G, red is Malbac, and gold is the generic MDA. And that's going to be the pattern that's used throughout most of these slides. Generic might drop out in a couple of the experiments. What you can see is the non-amplified, pretty nice and uniform, right? Pretty, not a lot of spikes. There are definitely spikes, this always happens. But in general, it's pretty darn uniform, not a lot of up and down in the coverage. That's also true for TruePrime. You can see it's a little bit more spiky, but overall the coverage is pretty darn uniform, especially when you compare it to the Repli-G and the Malbac and the random primed TruePrime fake kit, I guess you could call it. You can see these are really, really spiky. Okay, and remember, everything for the different chromosomes is jammed into these small areas. So if you spread it out, you're going to see very high peaks and then very low valleys for these guys. Whereas here, it should more mimic the non-amplified control. So let's zoom in. So that was hard to read. You know, you can look at the paper, it's still hard to read. But the bottom line is, here's chromosome 3. And you can see here's the non-amplified, same order. Here's the TruePrime. Here's Repli-G, Malbac, and generic. Okay, again, 12 million read pairs analyzed. They binned them. They looked at average coverage in those bins as they moved across the chromosome. Pretty good. I mean, my goodness, this looks almost identical. Okay, there's a little bit of a spike here, lack of a spike here, but overall these spikes are here. This gap is here. The gap is true in all of them, but you can see how much more variable the coverage is in terms of depth in all of these other kits and technologies. Okay, and we'll get into more detail as we go through. So here's looking at another chromosome. So that was chromosome 3. This is looking at chromosome 3 again, but now zooming in. So that first view was everything. That was this top view. Okay. Now here they've zoomed in to about 25 million base pairs, and you can see pretty uniform here. No big spikes. No big spikes with TruePrime, and again, big spikes up and down here. You can see lots of big spikes with the TruePrime with the random primers inserted instead of Primase. These big spikes, and then when you have big spikes, you lose coverage in other areas. And so you can see the overall coverage in this fairly uniform area. It's pretty uniform, and pretty consistent, but it's much lower than up here, right? Because a lot of the reads are being sucked up by the very high peaks of coverage. Now here's the 25 million. That's just a repeat of that one we just talked about. And then here's zooming into this region, which is about 500,000 base pairs. And you can see uniform coverage with non-amplified, very similar coverage with TruePrime, and then again, spiky with Repli-G, spiky with Malbac, and now a lot of regions are starting to drop out. And same thing with the TruePrime with the random primers instead of Primase. You can see very low coverage and lots of dropout regions, and presumably all these regions out here are sucking up those reads, and that's why the overall coverage is so low. And again, remember, they're only using 12 million reads in these experiments. That's to try to show the differences between the coverage. It's not enough to cover these chromosomes perfectly. So it's a way to give you an illustration that's interpretable and clear. Okay, so now this figure is looking at coverage in terms of percent of each chromosome covered. Okay, not depth, but percent. So there's got to be a read. If it's 100%, every base pair in that chromosome has at least one sequencing read over it. Okay. Again, 12 million read pairs were analyzed and then plotted for non-amplified TruePrime, Repli-G, and Malbac. And you can see, here's the gold standard. You can see that TruePrime actually had a couple bad chromosomes or underperforming chromosomes. Chromosome 19 and chromosome 22 actually didn't have nearly as good a coverage. They were actually undercovered compared to the non-amplified sample. And this is pretty consistent. We don't know what the reason is. You can look at the paper. They looked at very different variables, and I don't think we know what the answer is for this coverage. There's something unique about those chromosomes that make them more difficult to cover. Same thing with Repli-G though, and same thing with Malbac. Okay, and in fact, you can see the overall coverage of each of these is dropping. And we'll look at actual percentages with deep sequencing in a couple slides or the next slide. Okay. All right. Now, what about reproducibility? That earlier slide, I mentioned that people thought that MDA methods were low reproducibility. That is, replicates wouldn't perform very similarly. So the paper has in it an experiment to look at reproducibility. Okay. And so here's the non-amplified sample, and here's 4 different single cells amplified with the TruePrime single cell kit. And of course, everything's sequenced. Okay. This is analyzing 5 million read pairs across all the chromosomes. You can see again that the pattern is very similar between non-amplified and the replicates, and then that the replicates look very similar across all 4. Okay. It's hard to see here, so let's zoom in a little bit more as well. So here's looking at chromosome 4 in some detail. Here's non-amplified, the gold standard, the control. Here's replicate 1, 2, 3, and 4 amplified with TruePrime and sequenced. You can see this gap is here. These big peaks, these overrepresented areas are there. There's one peak here that's a little bit higher with TruePrime. But overall, again, across the whole chromosomes, very similar results, and very similar results within the 4 replicates. Right. And we're going to zoom in a little bit more, just one slide to show you, to prove to you how uniform this coverage is. Here's chromosome 6. Here is looking at this coverage again. This is the whole chromosome 6. It's different than the one I just showed you. And you can again see very similar patterns across the replicates and a very similar amplification pattern compared to the non-amplified sample. And then when you zoom into this region, I don't remember how big this is, but you can see nice uniform reproducible amplification with the WGA kit samples. And then in the non-amplified, there's this region that's a little bit bumped up in coverage for some reason, and that's repeated in the WGA sample. So whatever's going on here, the TruePrime kits are reproducing that as well. So if there's maybe some copy number variation or something like that going on, it's doing a good job of recapitulating that in the samples. Okay, so that was looking at 12 and 5 million read pairs. Now let's look at some deep sequencing results. And again, this is in the paper. If you want to dig into this a little bit harder, please go for it. Non-amplified control, TruePrime, Repli-G, and Malbac. And this is looking at over 500 million reads, okay, for all the samples. Coverage is about 20x, okay, for depth, okay, there and there, okay. But then what you can do is you can start to look at the areas that aren't covered. Remember I mentioned that if you've got these big spikes, those reads are being used up. When you sample reads, they're being used up in the big spikes. So you're going to have bigger valleys. And what that means, if the valleys are big enough, you'll actually have a lot more areas that aren't covered. And in other words, they're zero coverage regions. Okay? And so let's look at that in a little bit more detail. So for non-amplified, there were about 77,000 non-covered regions, a little bit higher with TruePrime. So there were 173,000 areas not covered with TruePrime. But then when you move over to Repli-G, it's up into the 1.2 million and 2.7 million for Malbac. Okay? And this really is what you'd expect based on that up and down spiky coverage that we looked at in those previous figures. Minimum length, 1 base. So there were regions where only 1 base was not covered, and a region of 30 million bases that wasn't covered, or base pairs that wasn't covered. So this looks like it's either maybe something that's not present in the 293 cells, or a truly problematic area, either for the library kit or for any of these methods. Probably for the library kit, because they're probably overlapping. And then mean length. So these are the number of zero regions. This is the average length of each of those regions. So 3.3 kb for non-amplified, and then 1.5 kb. So there were more of them with TruePrime, but they were shorter. And there were fewer of them with non-amplified, but they were bigger. So when you multiply those together, and you look at the total length of zero coverage, they're very similar between non-amplified and the TruePrime sample. So this is good, right? You're getting a very good representation of the whole genome in the TruePrime amplified sample. And that translates to coverage of about 91.6% for non-amplified and with the TruePrime, 91.26%. So they're just off by this, what is it, 12 million bases that weren't covered in TruePrime versus the non-amplified. Okay, so really good coverage. However, not the case with Repli-G using the random primers commercially available kit or the Malbec. So here you're jumped up to almost 500 million bases of no coverage, and here 1.2 billion, 1.3 billion bases of no coverage. That's almost a third of the genome. So when you look at coverage as a percent of the genome, it's 86% for Repli-G, pretty darn good, but still not as good as TruePrime or the non-amplified. And then Malbec is only around 60%. Okay. Finally, down here at the bottom, they looked at error rates, and they said that there were really no statistically significant differences in error rates between the different methods. So that's good. All right. So what does this really mean in terms of how does good coverage, uniform coverage, or bad coverage, or non-uniform coverage, either in breadth or depth, how does that translate to the experiments that I want to do or that you want to do? So we mentioned that one of the first things you might want to do is copy number variation. You want to look at, let's say, a cancer tumor sample, and you want to ask, is there any copy number variation going on that might be associated with the phenotype or the severity or whatever with this particular tumor or tumor cell? And so in the paper, they used the deep sequencing results. They calculated the number of reads in 500 KB bin sizes. So basically, the more reads in a region, the higher the copy number of that region in the genome. Okay, and then they deduced the ploidy level by Ginkgo analysis. That's the program they use. They use another one that's also in the paper. This was from the supplemental data. This data is a little bit easier to look at. That's why I use this one. The other one was a circular plot in the paper. But the bottom line is the more reads, the greater copy number, and vice versa. So the fewer reads, the lower the copy number. And so what Ginkgo did is it assigned copy numbers to the various chromosomes. So here you're working your way across from chromosome 1 all the way out to Y, okay, in this sample. Each of the grays vertical stripes is a different chromosome. And don't ask me why, I'm not a bioinformatician, but the non-amplified sample, the baseline was 3.1 copies. Okay, so 1.5x a normal diploid genome. I think this is just an arbitrary number. What I want you to do is compare the patterns across these different experiments. Okay, so non-amplified, you see nice tight bins, right? The number of reads is very tight, which you would expect with a non-amplified sample, and then you can see some copy number variation. Quite a few insertions or duplication events or triplication events that are above the baseline, and then some deletions as well, represented by the blue, that are below the baseline. When you look at TruePrime, it looks very similar. It's not perfect, but the bins are very tight again. They're a little bit wider than the non-amplified, but again, that's exactly what you would expect from the coverage type data that we looked at in great detail, right? The coverage is not as tight as non-amplified, but it's very tight, and it enables this program to make these C and V calls, okay? Whereas down here, you can see the scatter. So Repli-G, Malbec, and generic. The scatter is much, much greater, and so Ginkgo has a much harder time making copy number calls, and that's obvious. As you can see, the patterns are all over the place. They're not nearly as similar to the non-amplified as the TruePrime. And then finally, what about SNVs and ADO, or allelic dropout? Okay, so what they did was first they calculated SNVs, so the number of single nucleotide variants versus the reference genome, human genome, and they came up with 3 million for non-amplified in the deep sequencing, 2.7 for TruePrime, 1.7 for Repli-G, and then for Malbec, 2.55 million SNVs. So large numbers of SNVs. They obviously varied by method of amplification. What's the median overlap? So how many SNVs overlap with the non-amplified? So of course this is a one-to-one because it's the same sample. 2.4 million overlap here, 1.7 overlap here, and only 0.8 million here. And that translates to 80% overlap, 45% overlap for Repli-G, and 30% overlap. So the TruePrime kit was much better at detecting SNVs that were also detected in the non-amplified sample. You look down here, this one's kind of interesting. Malbec had a whole lot of SNVs, but a low overlap. I suspect that these are probably PCR errors because, again, it basically uses a PCR step at the end of the protocol. I'm guessing, educated guess, that these are probably PCR-induced errors, and that's why they don't overlap. Then they looked at SNV conversion, so heterozygous to homozygous conversion, or ADO. And this is basically if you have allele A and allele B, one of those alleles for some reason is preferentially amplified during WGA. And so you lose that other allele in your analysis, in your final sample. And so here, they again use like 4 different methods, okay? And it varied dramatically depending on the method that they used to do these calculations. Same here as well. But the patterns were basically the same. So this is the median results presented. So TruePrime only showed about 6% conversion, Repli-G about 30%, and Malbec about 31% of the SNVs that they could identify. Okay, and so take home is that you can definitely make good SNV calls, at least 80% overlap with non-amplified, and I think we could even improve on this in the last slide, as I'll talk about. But 80% is really good, and then this very low conversion rate, suggesting that there's very little bias in what the TruePrime kit wants to amplify. So it'll amplify both homologous chromosomes, and it'll do it fairly equally so that both alleles are represented in the sequence sample. All right, so just to conclude, I think we showed that the sequencing breadth of coverage nearly identical between TruePrime and the non-amplified sample, okay, both about 92%. And it's significantly better than Repli-G and Malbec, which were 86 and 59% respectively. Also more uniform sequencing depth, that parallels the non-amplified sequencing the best. That was very obvious with the spikes and the heights and the valleys of the coverage analyses, depth analyses. And then SNV calling is definitely possible with the TruePrime amplified samples, so 80% overlap. And then decreased ADO or allelic dropout with the TruePrime kit compared to the other technologies. So then, of course, I mentioned that these are kits that were developed by a company named Sygnis. We distribute them in the US. We can also distribute them in some other areas as other countries as well. So if you're interested in getting your hands on one of these kits, if they're in the US, you can go to our website. Prices and catalog numbers are here. Order them up. If you're outside the US, definitely contact our customer service team and get their, they can help you either order them or direct you to the right group at Sygnis to place your order. And then finally, this is the one last thought I wanted to leave you with. We're almost out of time, but as I mentioned, that NEB kit that's being used actually has a PCR amplification step at the end. There's lots of benefits to that, right? It makes it easier to figure out how much material to load on the sequencer and so forth. But the bottom line is WGA, as we talked about, might have bias depending on the kit that you use. And then the PCR step of the library might introduce some bias. So what if you could do a PCR-free library at the end instead of a PCR plus? And so Sygnis and we're working together to collaborate on an experiment like this right now. We've got our NextSeq AMP-free kit. This is a PCR-free kit, requires only 75 nanograms of sheer DNA input. WGA with TruePrime will produce a ton of DNA, much more than we even need. And this kit is the most efficient library, so it creates the highest percentage of fragments in the library with adapters at both ends that are sequenceable. So if you combine that with this WGA TruePrime technology, I think we could actually make those SNV calls better, the CNV data better, and so forth. So just wanted to end with that final thought. And we'll take any questions now. If you don't get your questions answered today, absolutely contact our tech support team. You can contact me if you want. I'll just probably have to get help from tech support. But absolutely reach out to us. And I want to thank you for attending and also our friends at Sygnis for providing this great data that we could work with.
If you have additional questions, if we didn't get to your questions, you can call or email our tech support team at the email address or phone number listed on your screen. Also, you will be getting a follow-up email from us with the link to the webinar slides and the webinar recording. And we can try and go through our chat window here, and in that communication we can try to answer some of these questions that we didn't get to today. A lot of great questions, a lot of great feedback from our listeners, so thank you very much. So keep an eye out for that follow-up email. I'd like to thank you all for joining us today and thank our speaker, Rob Brazos. We had fun having you. We hope that you learned something along the way today with us, and thank you for joining us. We're going to go ahead and close the webinar.
Thanks, everybody, and enjoy the rest of your day wherever you are. Take care.